Dr. Sage here, continuing our discussion of the host defenses. In this video, we'll discuss step 2, which is a presentation of antigens. Once again, this is a very brief overview. For a more detailed explanation, please take an anatomy and physiology course. By the end of this video, you should be able to compare the terms antigen, immunogen, and epitope, list characteristics of antigens that optimize their immunogenicity, describe how the immune system responds to alloantigens and superantigens, and list the types of cells that can act as antigen-presenting cells. An antigen is a substance that provokes an immune response in specific lymphocytes. To be perceived as an antigen or immunogen, a substance must meet certain requirements in foreignness, shape, size, and accessibility. Antigenicity is the property of behaving as an antigen. Antigens must be perceived as foreign. For example, whole microbes or their parts. Cells are substances that arise from other humans, animals, plants, and various molecules. Molecules of complex composition, such as proteins or protein-containing compounds, are more immunogenic than repetitious polymers composed of a single unit. Chemical categories of antigens include proteins and polypeptides, lipoproteins, glycoproteins, nucleoproteins, polysaccharides, and lipopolysaccharides. A substance must be large enough to catch the attention of surveillance cells to initiate an immune response. Generally, large antigens are better than small antigens. Molecules with a molecular weight less than 1,000 are seldom complete antigens. Molecular weights of 1,000 to 10,000 are weak antigens, and molecular weights of 100,000 the most immunogenic. Epitope is a portion of an antigen molecule recognized and responded to by our lymphocyte. It's the primary signal that a molecule is foreign. Haptens consist of only a determinate group. They're too small by themselves to elicit an immune response. If they're linked to a carrier group, the combined molecule develops immunogenicity. Alloantigens are proteins and other molecules of one person that are antigenic to another. Cell-surface markers that occur in some members of the same species but not in others. The basis for an individual's blood group and major histocompatibility profile. It's responsible for incompatibilities that occur in blood transfusion and organ grafting. Antigens must be further acted upon and formally presented to the antigen-presenting cells, which are macrophages, B cells, and dendritic cells. As an example, a bacteria is engulfed by phagocytosis into a dendritic cell and is encased in a phagosome. Lysosomes then fuse with the phagosome to digest the bacteria. Immunodominant epitopes are associated with major histocompatibility complex 2 and presented on the cell surface. Once again, this has been a very brief overview of the host defenses, step 2, presentation of antigens. Until next time, this has been Dr. Sage.